Good morning and welcome, everyone. So it's my pleasure this morning to introduce Dr. Jan Abkowitz to speak this morning. Dr. Abkowitz is a Clement A. Finch Professor of Medicine in the Division of Hematology and serves as the Division Head of Hematology here at the University of Washington. She is an adjunct professor in the Department of Genome Sciences and currently serves as the Associate Director of the Hematology Malignancy Program. Dr. Abkowitz earned her MD at Harvard Medical School, went on to complete her internal medicine training at Beth Israel Hospital, and subsequently completed her fellowship training in hematology here at the University of Washington. Dr. Abkowitz's achievements in her role as a clinician, as an educator, and a medical researcher are numerous. As a clinician, she has been recognized several times as top doctor in Seattle Metropolitan Magazine's annual survey, as well as nationally by US News and World Report. As an educator, she has taught at the undergraduate and graduate level and served as an instructor to her peers at the American Society of Hematology and the European Hematology Association. As a scientist, her research is focused on the molecular and cellular events that control red cell differentiation, especially how these cells grow and how their differentiation fails in diseases like aplastic anemia and myelodysplasia. These efforts have led to over 100 manuscripts, multiple book chapters, and innumerable national and international speaking engagements. It is with this background that she comes to us today. Please join me in welcoming Dr. Jan Abkowitz as she presents myelodysplastic syndrome and anemia.
So thank you for that lovely introduction. So today I'm going to be talking about myelodysplastic syndrome, or MDS, and anemia. And I have no financial disclosures. I will speak about a novel indication for differentiation, which is an iron chelator, as well as briefly about investigational studies of eltrombopag, melistat, and lusterapep. But I actually do have a disclosure, and that is that I have a bias. I'm an absolute believer in bidirectional translation, and the intent today is to convince you that that's a super goal for a physician scientist. So the concept is that you don't just bring things from the laboratory to the patient, but you're actually a clinician, you observe patients, you raise physiologic questions, you go to the laboratory, you do investigation, you go back to the patient or patient material with a new physiologic question and move on. And that this reiterative process is what drives, at least physician scientists, and makes, for me, life really fun. So I want to present a patient to start. This is Mr. DB. So Mr. DB is a 68-year-old with fatigue that interferes with his daily activities. He has many complicating medical issues, including type 2 diabetes, mild renal failure, hypertension, he's status posted in STEMI, he has paroxysmal atrial fibrillation, and sciatica. His medications include diltiazem, amiodarone, and apixaban. And I suspect he looks quite familiar to anyone who practices adult medicine here, because these are the common comorbidities of a 68-year-old or someone older. And so because he has fatigue, you get a CBC. And the CBC shows that his white blood cell count is normal at 5.8 with a normal differential. His platelets are also normal at 400. But his hemoglobin is 9.1, his hematocrit is 27, his MCV 106. So he's anemic, contributing to his symptoms. And so you appropriately remember your second year course, or now first year course, and say, I better get a kit count and figure out whether it's a problem of production or whether it's a problem of destruction. And so you get a reticulocyte count and it's low, so his marrow is not making erythroid cells. And then you say, wow, this is someone with mild renal failure, creatinine of 1.8. Maybe his kidneys don't work and there's not enough drive to his marrow, his erythroid marrow, to make erythroid cells. And so you measure erythropoietin levels, since erythropoietin is actually the only true hormone in hematology, because it's made by the kidney, goes to the blood, and ends up in the marrow saying, make more red cells. But his erythropoietin level is appropriately high, or close to appropriately high, for his degree of anemia. So his kidneys sense the anemia and are functioning perfectly to say, try to make more red cells marrow. And then you kind of say, oh, wow, the MCV is 106, and I seem to remember I should send a B12 and folate. And so you send that off, even though the folate is a totally worthless study in this setting, and they come back normal. So you send this patient to a hematologist who marrows him. And the marrow, too, is normal with a myeloid to erythroid ratio that's 2:1, with less than 2% myeloblasts, so an absolutely normal-looking marrow. The ratio of myeloid to erythroid cells ranges from 2:1 to 4:1 in normal marrow. But then cytogenetics come back. And the cytogenetics come back with a deletion of chromosome 5q in 12 of 20 metaphases. That's confirmed by interface fluorescent in situ hybridization and leads to the diagnosis of MDS. So what is MDS? So it's a neoplastic clonal process. So by virtue of having the 5q deletion in multiple cells, you know that there's a clone that's expanded. So the cytogenetics essentially absolutely defines that this patient has MDS. Sometimes the diagnosis can be more tricky because the physiologic definition is what's in that top line, that MDS is a neoplastic clonal process that initiates in a hematopoietic stem or progenitor cell and results in cytopenia. And so you have the cytopenia, that's pretty easy. But to show that it's neoplastic and clonal sometimes can be more tricky. Cytogenetics, of course, is a very clean way of doing that. The median age of diagnosis is 71. It's approximately 80% of MDS patients are diagnosed because of anemia, and it's a major cause of morbidity. So as we think about this patient, he's on a calcium channel blocker, he's on another vasodilator and amiodarone. Orthostatic hypotension is going to be accentuated by any anemia. He has sciatica, so his balance may not be right, and he's also on apixaban. He's anemic with poor balance, he's going to fall and he's going to bruise. So there's multiple connectors to anemia, such that it's actually part of every description of frailty in numerical ways of looking at frailty and its impact on clinical care, particularly in the elderly population. But the unusual thing here is that the anemia is generally macrocytic, which means the red blood cell size is big, and it's uniformly due to ineffective erythropoiesis, which is what we saw, meaning that you can see erythroid cells trying to develop in the marrow. They were there at a ratio of, actually a high ratio of 2 to 1. But the retic count wasn't coming out. So there's failure during the differentiation process in the marrow. And that's one of the real hallmarks of MDS, that there's ineffective erythropoiesis. So here is a list of some of the clinical associations of anemia in older persons: increased falls, increased rate of major depression, increased rate of hospitalization and longer duration of hospitalization, decreased mobility, decreased bone density, skeletal mass, and increased mortality in the Framingham studies. And so you can appreciate that anemia is really complicated by comorbidity when it occurs in the setting of MDS with a median age of 71. So just a quick back step into how a hematologist might start to think about this problem and think about it physiologically, which is where I'm going to divert to. So on the left there we go, there. So that's a normal red cell smear. Red cells are nucleate sacs. Right? Their only purpose in life is to bring around, they have hemoglobin to bring around oxygen and bring back CO2. And they are deformable, so they can get to small capillaries, and they can do that very efficiently and very, very well in any physiologic setting. So normally, the normal size of a red cell, or normocytic red cell, has a mean cell volume of 80 to 100. It's normochromic, which means if you stain it with the stain here that stains hemoglobin, and so it's red, that has a normal amount of hemoglobin, so its mean cell hemoglobin content is normal. And the most important thing about a red cell to remember physiologically is it tries to keep its mean cell hemoglobin concentration the same. Because if you have a concentration in a particular way, then you can optimally deliver oxygen, you can optimally bring back CO2. So there's many, many physiologic drivers, intrinsic to the red cell and extrinsic to the red cell, that tries to keep the MCHC, or the mean cell hemoglobin concentration, equivalent. And so, we still do look at smears. And when you look at the smear, 1/3 red, 1/3 white, 1/3 red, defines that more or less as normochromic and with a normal MCHC. And so that's a normal smear. Now, what we see perhaps more commonly is a smear on the right, which is microcytic, so a small MCV, and there's a reference point there, which is the small lymphocyte. And a small lymphocyte has a nucleus of 8 microns, which is the size of a normal red cell, across the normal red cell. And one can see that these red cells are smaller, and they also have more white, so it's 1/3, lots more white, not 1/3 red. And so they're microcytic hypochromic. And so they don't make enough hemoglobin. Either they have a failed heme production, which the most common reason for is iron deficiency, or they have a failed globin production. The most common reason for is thalassemia or thalassemia trait. And again, the take home I want is that cells try to maintain a normal MCHC by making smaller cells. So the very first thing that happens in iron deficiency is that you make smaller cells to try to compensate. And so the smaller, so you retain your MCHC, and then when you can't do that anymore, you become anemic. So what about this patient? This patient is macrocytic. The MCV is greater than 100. It was 106. There are, on smear, some hyperchromic, more than 1/3, 1/3, 1/3 red cells, and certainly some big red cells. And so, though we don't think about it this way as much as hematologists and as clinical hematologists, and I see this clinical hematologists in the crowd, when I thought about this in the context of physiology, the next question was, are the cells made bigger and then make more hemoglobin as a compensation to try to maintain their normal MCHC? Or is there excessive heme or excessive globin, and the compensation is that they're made bigger in order to try to maintain an MCHC when something's awry, making too much heme, for example. So what I plan to do today is to present studies of MDS resulting from an isolated deletion of chromosome 5q, patient that I just presented, and of a congenital macrocytic anemia that's phenotypically quite similar, called Diamond-Blackfan anemia, but again, congenital, so associated, as you'll see, with mutations that are inherited. I'm going to show that these two very disparate anemias in terms of their pathogenesis, or in terms of their initial thought about pathogenesis, actually result from a common pathway. They result from excess heme in the cell. I'm going to argue why this physiology is more generalizable to actually most MDS anemia, and therefore provides new insights for therapy. And as importantly, I'm going to show how different technologies such as human marrow cell cultures, mouse models, quantitative flow cytometry, and single-cell RNA sequencing can be combined to answer physiologic questions that have clinical impact. So first, a little bit about DBA, Diamond-Blackfan anemia, and del(5q) So as I alluded to, Diamond-Blackfan anemia is a congenital macrocytic anemia. 25% of DBA patients cases result from haploinsufficiency, so a mutation that causes a loss of function of one of the ribosomal protein S19 genes, and 65% from haploinsufficiencies of 18 other ribosomal proteins. So haploinsufficiencies of 19 ribosomal proteins result in the same phenotype, and that is erythroid marrow failure. And the macrocytic anemia del(5q) what our patient has actually results from haploinsufficiency of ribosomal protein S14 that's acquired. So both a congenital mutation and an acquired mutation in a ribosomal protein, we're now up to 20 different ones, have the same phenotype, which is a failure of erythroid cells to differentiate, a macrocytic anemia. And when tested, these mutations disrupt ribosomal assembly and impair protein translation, which is a bit of a first clue, and it led most of the world to say, and the dominant hypothesis to develop, that if ribosomes don't assemble right, then there's stray ribosomal proteins around, and they're going to irk the cell. It's going to start the p53 activation pathway and lead to apoptosis. And so that was a common explanation for this physiologic finding. But the problem is, if this is true, why should red cell differentiation be impacted and not other lineages? Because the same thing in a congenital situation, and actually also in an acquired situation, when it acquired at the level of hematopoietic stem or progenitor cell, is in all lineages. And also, why are the cells macrocytic? And I've also given you a little bit of a clue that I'm going to link the concept of preserving an MCHC to that. So somewhat about erythropoiesis. Erythropoiesis is actually very cool to study as a basic scientist, because it's the biggest, fastest production of cells in the body by orders of magnitude. And so anything that has to do with transcription, translation, any process of replication is going to read out, at least in the erythroid series. And so it becomes a really, and it's also, red cells are accessible, bone marrow is accessible, so hematologists have advantages looking at erythropoiesis and its outcome. So we all produce 2.3 million cells per second. And since the reticulocyte count can increase 5 to 10% in response to anemia, this is very dynamically regulated. So we can make 5 times more or 10 times more and then turn it off and do it with fidelity and do it night and day over and over again. So it's exquisitely regulated, humongous production of cells and makes, therefore, a lot of fun to figure out why. And the second thing about erythroid cells that are kind of interesting is they only, at the end point, make a single protein, hemoglobin. Actually, it's a single protein globin that combines with heme, which is a chemical chelate. And so over 95% of the red cell protein content is hemoglobin. That's 270 million hemoglobin molecules per cell, each with 4 heme moieties, and now each with 2.3 million cells being made a second. So extraordinarily large quantities of heme are needed, and needed quickly as red cells mature, but red cells have a problem because heme's toxic. It leads to lipid peroxidation, Fenton reactions, extra reactive oxygen species. And so how does the red cell handle this kind of growth, this kind of need, and making hemoglobin with the fidelity that it needs over time? So here's the dilemma for the cell. Heme's a chemical chelate. It's assembled enzymatically. Globin's a protein, so it's produced by transcription and translation. Thus, co-regulation of heme with globin can't rely on the coordinate regulation of transcription and translation, which is the way most coordinate processes that we study in biology happen. And erythroid cells need an alternative solution. So this is going to be what I'm going to tell you about in all the subsequent slides. This is how red cells solve it, why it's relevant to our patients, why it leads to new ideas for therapies. And I'm just going to bring you through this slide slowly and then show you all the evidence with different methodologies that speak in part of it. So under the influence of GATA1, which is a transcription factor that initiates erythropoiesis, the erythropoietin receptor is upregulated, which is shown in green there, at the very earliest stage of erythroid commitment, called the BFUE stage. And then as further upregulated as BFU becomes CFUE, that next stage of commitment. What happens then is that erythropoietin, as well as GATA1, upregulates the transferrin receptor. Transferrin receptor is the receptor for iron that brings iron into erythroid cells. So about the CFUE proerythroblast stage, iron is entering erythroid cells, as shown there by the orange, and also the yellow, which is a CD36 marker, which is a good surrogate for CD71, which is a transferrin receptor, because they go essentially the same places. And at that stage of erythropoiesis, iron enters cells, right here. So iron enters cells, it binds iron-responsive proteins, and it induces the translation of ALS2, which is ALS2 is the first and rate-limiting step of heme synthesis. So as soon as iron's available, heme synthesis is made, or heme is made. And it does so by removing inhibitors, so it does so by inducing the translation of the initial rate-limiting step, and it does it rather quickly. And so as shown below, heme synthesis intensifies. And then heme is the inducer both of transcription and translation of globin, and does so by inhibiting inhibitors at both steps. And so globin is poised to be made, and as soon as heme is there, it is briskly made. And this is how coordination happens across differentiation. And one can look by many ways at the phenotype of these cells as they differentiate. From this stage onward, they're morphologically definable within the marrow. And one can also define them by the presence of CD36 or CD71 on the early cells, glycophorin A, which is a late marker, coupled with CD36 on some of these middle cells, and glycophorin A alone on some of these later cells, something I'll use later in defining cell types. So globin synthesis intensifies, but what's the problem? There's a period of time when heme exceeds globin. Now, red cells don't have it in their interest to destroy heme with hemoxygenase, which is how most cells control heme, because a second later, nanosecond later, some small amount of time later, they need that heme. So what they do is they export it to an exporter called FLVCR. And this, an inadvertent cloning of this exporter is what actually got me into this whole area of investigation. What FLVCR does is it exports excess heme and protects CFU and erythroblasts, proerythroblasts, from heme toxicities. And then when globin's there, it doesn't compete with globin, so globin is ready and able to take the heme and run with it. And so we've used this concept in this physiology, some of the initial papers are down below, to use as a tool, as well as just a tool to understand disease and a tool to understand biology, as well as just some insights. So what happens then? In normal times, shown above. FLVCR is there, and out goes heme and globin, out goes heme until globin is ready. But what about DBA and del(5q) MDS? There's poor ribosome assembly. It impairs globin translation. Heme is made pretty well because you just need very few enzymes, very few proteins to have an enzymatic process happen. So a little bit of translation makes a little bit of protein, you make plenty of heme, but you can't make enough globin, and you can't make it enough quickly. And so the time where heme exceeds globin is now big, and FLVCR is insufficient, and cells die of heme toxicity. And so that's the linking physiology I'm going to talk about and some of its implications. Okay, so the experimental design was to culture marrow cells from patients with diamond-blackfan anemia, marrow cells from patients with del(5q) MDS, normal individuals and MDS controls, and follow their erythropoiesis in vitro, where you have more control over the process. But remember, these are normal primary cells. These are not cell lines. And shown on the left is a DBA patient, as an example. And this DBA patient is a 28-year-old woman who had diamond-blackfan anemia diagnosed when she was 2 months old. She was initially responsive to steroids, which is pretty common in DBA, but lost that response, and is now transfusion dependent. So she's on no medications, and she requires transfusions to keep her hematocrit up, but her erythropoiesis in her marrow would be rather unperturbed. So an excellent example for how to look at the situation. And when one looks at the marker CD34, which marks the earliest progenitor cells, she has a lot of them in red, as does the normal on the right side. So there's plenty of progenitors here, and there's plenty of early progenitors there. But normally, this is the transferrin receptor, CD71. As cells mature, they pick up CD71, and then they lose the earlier cell markers, and they go through a pattern that looks like that. That's normal. But what happens in this patient? As soon as the cells turn blue, as soon as they have CD71, they die. So as soon as she's bringing iron in, her cells die. So this is the first piece of just direct evidence that perhaps this hypothesis about heme excess causing toxicity is true. This is clearly the stage of differentiation where things go awry. Now, if you look at this is another patient, and this is going to be the typical way I show a lot of things, CD36 on this axis, glycophorin on this axis. So differentiation goes from stage 1 to 2 to 3 to 4, and DBA from 1 to 2 to 3 to 4. But if you look quantitatively, and you can pick it up in the flow cytometry as well, and say how many cells are in stage 1 compared to stage 2, compared to stage 3, compared to stage 4, I want to point out two things. First, between stage 1 and stage 2, there's a drop-off. So just at the level we're looking at, the CFUE pro-erythroblast level, cells die. But there's a second really important thing, and that is those cells that survive, that are present in stage 2, actually expand in number in stage 3 and stage 4 and increase. So this allows us in the lab to ask two questions. First, what are the issues that happen at stage 1 that makes those cells die? And then what is the compensation that allows cells to actually grow and become stage 3 and stage 4? And so now are the results from the multiple patients, from 5 normals and 3 DBAs. Cell expansion is low in DBA. If one says, what's going on in stage 1 that makes cells die? There's a high heme content. And then if you say, what about those cells that make it to stage 3 or 4? What compensation did they do in order to make it through the differentiation scheme? Either they have less ALS2 RNA, so they make less heme, or they have more FLVCR RNA, they export more heme. So this becomes circumstantial evidence that the heme axis and that hypothesis that I told you about is indeed correct. And if you look at the MDS patients, the same thing is true. Little cell expansion, heme content high. Those that make it, ALS2 is low, or FLVCR is actually 10-fold higher. So they compensate in order to make it through. Now, there's always some problem, and I'm just going to talk a little bit generically now, with relying on flow cytometry, particularly a couple markers to identify a cell's type or stage. And the concept goes something like this. The flow cytometry just marks a surface feature on the outside of a cell that you can easily discern, but it doesn't necessarily say what that cell is thinking or what that cell is doing, or who that cell is. And so this is just a plug to be a little suspicious of any data that ends with a flow cytometry initial description. And it's something like this. So your leader, Dr. Brody, she has a red sweater on. If we had antibody to red sweater, she would be positive today. But tomorrow when she has a blue top on, she's the same Dr. Brody, but she's not going to read out positive in our assay. And that's the thing you have to keep in mind, okay? If you're going to think about flow cytometry as you read out. And so we wanted some additional ways of using, particularly our cultures, to ask the same question. And so what is done here is we just sampled the same culture where you put the marrow cells and you gave them the right ingredients to be able to grow over time. And we accessed them at day 3, day 7, day 10, along the way. And what I'm showing you is just the protein results. So here's DBA in blue, and here's normal in black in this example. And in this example at day 3, already in culture, heme is being turned on because ALAS2, that first and rate limiting step, is present at the protein level. And actually, as anticipated, there's even more here, heme turned on in the DBA. So the enzymatic process is intact that needs the small amount of protein that turns on heme. However, at day 10, the DBA is lacking the ability to make enough protein, globin. And so there's a discrepancy between heme present at day 3 and at day 10 in culture, globin present in abundance in the normal, but low in the DBA. And so then we asked the question, what's happening in between? Because we knew cells were dying in between. And so we looked at day 7, and at day 7, there was increased reactive oxygen species and increased apoptosis in the DBA samples as shown on the right. This is the same study done with actually the patient that I presented, marrow. And at day 10, it's the same discrepancy. Globin present in normal, globin not present in abundance in the DBA. Lots of ROS at day 7, and lots of apoptosis at day 7. Now you can show cause and effect by, in science, probably one of the easiest ways or best ways to show cause and effect is to try to reverse the process by inhibitor. And so succinyl acetone inhibits the second step of heme synthesis. So by adding it to the culture, heme synthesis is not going to be as robust. And so as we interfere with heme synthesis, twice as good growth of the DBA and twice as good growth of the MDS cultures without any impact, or with actually decreased growth, but not significantly in normal. So in summary of this first part of the data, it demonstrates that anemia occurs with when the synthesis of heme and CFU proerythroblast exceeds its use in hemoglobin or its export via FLVCR. And I'm just going to preempt it by saying that in a mouse model, if we knock out FLVCR, we have the same phenotype. By extension, if you limit heme synthesis, for example, if you restrict iron, this could be a novel way to treat DBA and del(5q) MDS. And then the last thing to think about is I'm talking about MDS that comes from a mutation that causes a haploinsufficiency of ribosomal protein S14 and therefore impairs translation. But MDS has lots of diverse mutations, many of which impede brisk protein transcription or translation. Any one of them would lead to the same physiology. There would be a problem with the quick initiation of globin production, and heme toxicity can contribute more broadly to MDS anemia. And so from this physiology as it relates to a congenital disease in a subtype patient, we kind of learned something that might be important for all patients with MDS anemia. And again, that's the most common presenting symptom and the most common morbidity in the setting of MDS. And so this has led to a clinical trial that's just underway. Its PI clinically is Bart Scott with Jason Cooper, who's a heme fellow. And I'm doing ancillary studies, as is Brent Wood, who's a flow cytometer person. It's a single phase open label phase 2, single center open label phase 2 study of deferricerox, which is a iron chelator in patients with good prognosis and low stage MDS and anemia with a ferritin that's at least above normal. And the primary endpoint is looking at the proportion of patients in which there's erythropoietic hematologic improvement. I want to change gears a little bit and talk about the mouse model. So if you delete FLVCR in a mouse, if you do it congenitally, if you do it neonatal, if you do it in utero, they die. But if you make the mouse live and be born and then conditionally knock out FLVCR, they develop a macrocytic anemia that's absolutely identical to DBA and del(5q) MDS. So they're very anemic. Hematocrits there of 15, low hemoglobin, and a high MCV. So the macrocytic anemic, the exact same phenotype. If you do flow cytometry with a mouse, TER119 is a glycophorin A equivalent, and here's CD71, so differentiation proceeds like that. And they have a block between stage 1 and stage 2. Shows here, lots of cells here, accumulate because they're blocked, and they don't go onward. And so the block is at exactly the same stage, the CFUE proerythroblast stage. If you look at heme content, it's higher, shown in yellow here. And if you look at cytoplasmic ROS, it's higher. So the model is an excellent convergence of a knockout of a protein exporter, a abnormality of a ribosomal protein, etc. So it's a final common pathway that's conserved. And so here's the scheme again. GATA1 is now shown, so the transcription factor that starts erythropoiesis, up goes erythropoietin receptor, transferrin receptor goes up, iron comes in, iron binds to iron-responsive proteins and induces the translation of ALS2, which is the first and rate-limiting step of heme synthesis. Heme synthesis intensifies. Heme induces globin transcription and translation. There's a period of time where heme exceeds globin, and at that time, if you're lacking FLVCR, so you can't export that excess heme, cells die. So this becomes a really interesting tool to say, what does extra heme do to cells? How does it work? What's the physiologic and the pathologic consequence of extra heme? Because there's absolutely nothing else wrong with the transcriptional, translational, or otherwise programming of erythroid cells. They're just lacking the ability to get rid of heme at a particular stage. And so we began to use this as a tool to explore normal and pathological erythroid differentiation. And so here's the single cell experimental design. We identified early progenitors through flow cytometry that were in the stages between BFUE and basophilic erythroblast. We isolated single cells and then quantitated that cell's surface marker expression with flow, and then barcoded the cell and sequenced this total transcriptome. So for individual cells, we knew what they expressed at the surface and what their total RNA sequence content was, and could ask questions about the correlation of protein to RNA, and also questions about what happened in both pathological situations and normal situations. And we linked the surface protein expression of a single cell to its RNA-seq, which is indeed a somewhat novel method. So first, that these methods are reliable. So globin, it actually has two chains, an alpha and beta chain. They have to be made synchronously to make a hemoglobin molecule that has two of each. And they're on different chromosomes. So this is a true example of transcriptional regulation, translational regulation, because they have to be made coordinately. So indeed, there is quite spectacular coordination across all the single cells between the transcript measure for alpha globin and the transcript measure for beta globin. If one looks at the correlation of protein, which is the late protein TER119 and glycophorin A, there's a good, which is the gene, there's a good correlation, but not perfect, because protein and gene expression should be correlated, but there's other things that drive a cell to make protein in different quantities. And there's a very good correlation as well between CD71, the transferrin receptor protein, and the transferrin receptor gene expression. So recognizing this and doing what's called single-dimensional PCA, which I'll get back to in a minute, we realized that TER119, the glycophorin A equivalent, was really a very good distinguisher of cells at different stages of differentiation. And so in a very unbiased fashion, we said, take a look at our normal cells computer and distribute them across the amount of TER119 they have on their surface. And out came this, what's called a violin plot on the left side. And the violin plot made four different groups, A, B, C, D, we call them, less mature, more mature, by virtue of the TER119 expression. Now, the important take home here is if you do this in this kind of detail, people tend to say, well, there's TER low, there's TER high, and make an arbitrary cut into two across that. And it's a lot of arbitrariness. But by using an unbiased method, we actually separated cells into these four categories where there's different numbers of cells in each category, but they're driven by the amount of TER119 on the surface. And then did the same with what I'm going to show you, FLVCR deleted. We've also done erythropoietin controls, and we've done some RPL11 deleted mice and DBA mice, those of which I'm not going to show today. The first thing is that when one uses these exact same cutoffs that are derived from the control and applies them to the FLVCR deleted mouse, they nicely split them into categories. But category A, the very earliest cells, had two subtypes. Shown in gray there are dying cells. Those cells essentially had very few transcripts, period. And about 50% of the cells were dying. And so again, this method confirmed exactly what we know at stage A in the mouse. 50% of cells die because they have excess heme. And so that the readout, we were very excited about and very pleased with, that 50% of the cells had no readout and 50% had readouts that were very equivalent to the control. And so in subsequent analyses of transcriptome, we just excluded those AD cells because they would drive results in a wrong fashion. So when one looks at single cell data, one of the common things to do is to look at principal component analyses, or PCAs, which just looks at variance across a large data set. And the take home that I want here in this show of the PCA is in group A from the deleted cells and in group A from the wild type cells, they just overlap. They express genes in exactly the same way, because they are very similar. However, the wild type patterns through here, while the deleted patterns this way. And so from the PCA analysis, we knew that we were going to pick up things from the individual transcriptome analyses that were going to be interesting about how heme drives erythropoiesis, and then by extension, why when heme is in excess, there's a problem in erythropoiesis. And indeed, there were some amazing surprises. These were things that I'm going to show you now that solve the problem that we were looking at, but we actually absolutely didn't anticipate beforehand. These are not directed studies. So the next thing you do is you take 42,000 gene sets, and you say, what are different among these gene sets, these 42,000 gene sets between the deleted and the wild type? And the first thing that came up, and this is plots of variance, is that the FLVCR deleted erythroid precursor cells had high levels of RNA encoding ribosomal proteins. And particularly early, stages A and B, very high levels versus the amount that's seen in the wild type. And that was unexpected. It went across all the ribosomal proteins. But then we sat back and thought about it. You know, if heme's going to turn on globin and has to do it real fast, it's going to turn on transcription and translation of globin, but it better have ribosomes in order for globin to be translated. And so it made sense. The integrate to the heme circuit was to turn on ribosomal proteins. And it does it for the aficionados here in a non-GATA1 dependent fashion. I'm sorry, a non-BAC1 dependent fashion. And so we wanted to prove that was the case. And so went to early human marrow cells from normal person's marrow and said, if at time 0 we give ALA, which bypasses ALS2, the rate limiting step, so makes heme synthesis happen artificially, bypassing the rate limiting step of the cell. What happens 15 minutes later and what happens 30 minutes later? So what are the immediate responses to making a cell make heme? And indeed, the RNA shown in black and the protein for ribosomal proteins, including ribosomal protein S14 for what we saw in the del5q abnormality, and ribosomal proteins S19, the most common finding in DBA, and several of the others that are commonly found in DBA. So that this primary cell data absolutely confirms the physiology. Now, the last thing we did in this domain is said, okay, it's making lots of ribosomal proteins, but can they be adequately assembled? Is it making a ribosomal protein RNA? Is it making ribosomal RNA as well, so that ribosomes are actually made? And this is a reasonable surrogate for ribosomes being made, looking at ribosomal RNA. And indeed, by 30 minutes, there is ribosomal RNA being made in excess. So heme turns on production of ribosomes. The second surprise is here. This is the second thing, again, to just the most down. So that was the most upregulated. This is the most downregulated of the 42,000 gene sets that we looked at. And that's GATA1 and GATA1 targets, the major transcriptional regulator of erythropoiesis. So in A, in the cluster in A here, more or less the same. It turns on or turns on a little bit appropriately, but it goes sky high in B, C, D in controls and doesn't in B, C, D, and FLV-deleted. So it's more downregulated in the FLVCR deleted. And again, it was the most downregulated that came out of the survey. And when one looks into the transcription factor, the cluster of genes that it targets transcriptionally and how they're turned on or turned off, the same thing can be seen here by this plot or by a different sort of way of looking at it. In A, essentially the same, a real turn up of all the genes that have been shown through other methods to be true GATA1 targets in B, C, and D, but not in the deleted. So excess heme turns down GATA1. Okay, that would be the take home. So let's go to the primary cells, and particularly the later erythroid cells where this appeared to be prominent. Do the same experiment, add ALA at time 0, and look at 15 minutes and 30-minute readouts. And indeed, both the mRNA and protein of GATA1 are significantly down. So a quick experiment shows that result. Now in cells that are primary cells, there's a limited number of experiments that you can actually do because these cells are finicky and they're primary, they don't live forever, and a cell line gives you some added advantage. And so we wanted to confirm the study in a cell line called K562, which has erythroid features. And so if one looks at K562 cells and does the same experiment and gives them ALA at time 0 and looks at what happens at 15 minutes, I'm going to take you through the red. Up goes heme, up goes reactive oxygen species, down goes GATA1 protein, and it starts making globin. But if you add acetone as well, so you start heme synthesis at step 2, but you turn it off at step 3, so it doesn't happen, then you no longer have excess heme, you no longer have heme go up, you no longer have ROS go up, you no longer have GATA1 protein go down, and you don't turn on globin. So it becomes a very nice, nice take home. So what does this mean physiologically? And that is that we think that heme and GATA1 are actually the major co-regulators. We have now a bunch of other data in support of this that regulate normal erythroid differentiation. So we think of erythroid differentiation in a normal circumstance this way. GATA1 upregulates ALAS2, transcription, heme synthesis intensifies, it turns on globin synthesis and also ribosomal protein transcription. As erythropoiesis goes on along this pathway, FLVCR, the heme export protein, goes down. And that's because it's transcriptionally made only at the CFUE stage and earlier, and then it is lost through cell division as cells mature. And so by the end, you don't have very much FLVCR protein. In fact, you don't have any. Heme accumulates again, and it assures that GATA1 is turned down and that erythropoiesis appropriately turns off. And actually, if you overexpress GATA1 in a way that it can't be turned down, erythropoiesis fails because it can't turn off at this stage. And so it appears that that's what normal differentiation looks like based on our data. And so what happens here? When we have Diamond-Blackfan anemia, del(5q), or FLVCR deficient erythropoiesis, then heme accumulates early, it prematurely downregulates GATA1, and erythropoiesis prematurely terminates as one of the processes, and death occurs at that stage. So if you put it all together, the different kinds of ways that heme can impact the physiology of DBA and MDS. Genetic mutations in MDS directly or indirectly result in slowed transcription or translation. And in DBA, translation is slowed because of the ribosomal protein abnormalities, and therefore heme exceeds globin. The excess intercellular heme causes increased ROS and cell death, as I showed you earlier. It increases ribosomal protein synthesis, which would amplify ribosomal protein balance if it were present, and amplify nucleolar stress. It decreases the transcription of genes involved in mitosis, which is data I haven't showed you, which is the third thing that came out of our non-biased screen to cause macrocytosis. And so heme actually contributes to this mechanism to make sure that the MCHC remains appropriate for its concentration. And it decreases GATA1 in this situation of DBA and MDS anemia, resulting in the earlier premature termination of the erythroid differentiation program. So shown visually, poor hemoglobin assembly because of the abnormalities of ribosomes can lead to nucleolar stress, p53 activation, and apoptosis. That we also connected it to excess heme, excess ROS, more ferroptosis and apoptosis, so a synergistic badness along these two pathways in diseases like MDS and DBA. And also showed that that excess heme turns off GATA1, for premature or early termination of the erythroid differentiation program, all of which will transpire to make fewer red cells. And so what does it mean? Well, it actually means a lot of really optimistic things in terms of therapies that exist or are in development. So that if one decreases heme synthesis, which one can do with decreasing iron, but there's probably a number of other ways that might be more safe and more interesting that we're investigating. Or if one stabilizes this pathway, which lenalidomide is a drug that's able to do that, particularly in the del(5q) abnormality situation, that you should have better erythropoiesis. And then also, if you're able to make erythroid cells more plentiful through anything that turns on more erythropoiesis, then you should have more cells entering the pathway, less, same amount dying, more coming out the other end, and a benefit for patients. And that's exactly what many of the approved and in clinical trial studies are doing. So prednisone presumably stabilizes in this situation BFUE to CFUE, makes more erythroid cells. So more erythroid cells enter the pathway, more can come out the other side. Erythropoietin, you can use it in pharmacologic dosing, especially if someone has an intrinsic erythropoietin level that's on the lower side, less than 200, which was not our patient. And then it overdrives erythropoiesis. And so overdriving erythropoiesis will give you the opportunity to have more erythroid cells out the other side. The hypomethylating agents, since MDS has a whole bunch of other gene defects that affect the input to this pathway, hypomethylating agents are going to make for more erythroid cells coming into the pathway, therefore more erythroid cells coming out the other side. And recognize, again, these are going to be synergistic with anything that we do that decreases heme synthesis and makes the hemoglobin situation more correlate. And in clinical trials, there are a number of other things that may increase erythroid cells. The thrombopoietin mimetics, and particularly eltrombopag, which is an interesting compound. It's a small molecule which triggers thrombopoietin receptor stimulation, which is on early hematopoietic progenitor stem cells as well as on platelets. But the intriguing thing about eltrombopag, for reasons that are just weird, it actually is an iron chelator. So it may be particularly efficacious in this situation because you get 2 for 1. And that's why this is a clinical trial that we're equally interested in participating in. Imetelstat, or telomerase inhibitor, and then a new thing on the market, particularly for MDS with ring sideroblast, is the Luspatercept. And again, presumably the mechanism of that, which is not as well understood, is again making more erythroid cells that goes down the pathway. So it should be synergistic. And with that, I'll end. And this is a very old picture of my lab from a lab hike to Mount Rainier. And it's because these studies actually develop over a long period of time. I want to credit from the beginning Sean Quigley, who was a postdoc in my lab many years ago that inadvertently cloned FLVCR. We were looking at for a virus receptor that had the phenotype of making a cat have pure red cell aplasia. Thought when we cloned it that it was going to be a cytokine receptor. And when you looked at its structure, when you did an expression cloning process, and that's what came out, it was a transport molecule. It took another 4 years to identify that it really was a exporter of heme, which is his work. And then knocked it out in a mouse and looked at the phenotype, and Siobhan Keel was the first author in that science paper. And that was the basis, really, for beginning to have a tool and having an opportunity to do these studies. The ones that I presented today are mostly the work of Zantel Yang and Raymond Doty, and particularly Raymond Doty, and Adam Monday, and some of the later Western analyses. And these single cell studies are a very close collaboration with scientists at the Institute for Systems Biology that are listed below. And so thank you, and I'm happy to take any questions.